glutathione gets oxidized. Glutathione gets oxidized. So this is reduced glutathione, GSH, gets converted to oxidized glutathione, that is GSSG. You also know that oxidized glutathione is a dimer, that it is made up of two molecules of reduced glutathione, right? So two molecules of reduced glutathione gets converted to oxidized glutathione. On the other hand, H2O2 is getting destroyed, and this is a very useful reaction in RBCs, right? And here the enzyme is glutathione peroxidase. This enzyme is a selenoprotein. Selenoprotein means it requires selenocysteine amino acid at its active site. So answer to this question is selenocysteine, right? Also, this selenocysteine is present in many other enzymes, and all these enzymes are known as selenoproteins. I just told you. So let's take the examples of selenoproteins. Mainly they are reductases. And peroxidases. So one example we have done is glutathione peroxidase, but here it is not glutathione reductase. Remember this point here, this is not glutathione reductase. Glutathione reductase requires FAD, and glutathione reductase is the marker enzyme for B2 deficiency, because FAD means B2 we say, right? Now, second enzyme I'm going to tell you here is, so first is glutathione peroxidase, second is thioredoxin reductase. Thioredoxin reductase enzyme is involved in the conversion of ribonucleoside diphosphate into deoxyform, right? Deoxyform, because deoxyform is required for DNA. So we have to convert ribo into deoxyriboform. At that time this enzyme is used, that is thioredoxin reductase. This is also a selenoprotein. Then third is iodothyronine deiodinase. This is involved in thyroid metabolism, right? So these three are examples of selenoproteins. And now you know that selenocysteine is 21st amino acid. The precursor for selenocysteine is serine, right? All these are frequently asked questions about selenocysteine 21st amino acid, right? So let's take question number 2. In this, the question asked is, which is the only non-protein member of ETC? So let's first take all the members of ETC, right? You know that ETC occurs in inner mitochondrial membrane. So all the components are lying in inner mitochondrial membrane. We have 5 protein complexes. That is Roman number 1 to 5. Complex 1, complex 2, like that. We have 5 protein complexes which all are fixed within the membrane. See, all the components are present in the membrane, I am saying, and I am now saying that these 5 complexes are fixed in the membrane. They cannot move in the membrane, right? They are fixed proteins present, right? On the other hand, we have 2 mobile electron carriers. See, now these 2 components are mobile, means they can move within the membrane, right? And these 2 components are coenzyme Q and cytochrome C, right? So first is coenzyme Q, also known as ubiquinone. Other is cytochrome C, which is a kind of peripheral membrane protein. Right? So now note one point here, that cytochrome C is also a protein, right? And all these 5 complexes are also what? They are also proteins. So which is the only non-protein member of ETC? That one left is coenzyme Q. So answer to this question is coenzyme Q, right? So coenzyme Q is not a protein. It is it is lipid or carbohydrate. What do you think? You can note one point here, no need to cram it. You can note one point here, that this coenzyme Q is a component of ETC. It is mobile within the membrane. So anything which can move within a lipid bilayer membrane is Obviously a lipid, right? So it is a lipid which is hydrophobic, right? So coenzyme Q is the only non-protein member of ETC, right? Then next question, question number 3. Muscles cannot make use of glycogen because of deficiency of. Usually most of the students have tendency to mark glucose 6-phosphatase as answer to this question whenever they ask you muscle and glycogen. So you quickly mark glucose 6-phosphatase because of one basic point, that in muscles glucose 6-phosphatase enzyme is normally not there. That's why muscles cannot maintain blood glucose. See, because glucose 6-phosphatase enzyme is not there in muscles, that's why glucose 6-phosphate is not broken down to glucose. This enzyme carries this reaction. When in muscles this enzyme is not there, this reaction is not carried. So we do not get free glucose in case of muscles. So muscles cannot give this glucose to blood, right? This glucose 6-phosphate stays inside the muscle cell only and it will enter into glycolysis, right? But answer to this question is not glucose 6-phosphatase. Please read the question very carefully again. See the question again. Muscles cannot make use of glycogen because of deficiency of. Answer here is glycogen phosphorylase. Let me tell you, the other question is different. The other question is that muscles cannot maintain blood glucose levels because of deficiency of which enzyme? Then answer is glucose 6-phosphatase, right? But now the question is different. They are saying that muscle cannot use the glycogen. Means glycogen in muscles is not getting broken down. And what is the first enzyme to break down glycogen? The first enzyme to break down glycogen is this enzyme only, glycogen phosphorylase, which is also the rate limiting enzyme of glycogenolysis, right? So if this enzyme is not there, then muscles cannot use their glycogen, right? So answer is glycogen phosphorylase, not glucose 6-phosphatase. To this question, be careful in reading this question, right? Next question, question number 4. NADPH is used in. Usually students have a tendency to mark all for this question because you are confused between two things about NADPH. You have to be very clear that NADPH is used in which pathway. And you also have to be very clear NADPH is produced in which pathway, right? So let's be very clear. Answer for this question is not all. Answer is fatty acid synthesis only, because the question is asking NADPH is used in, right? So let's first see NADPH is produced in which pathway. NADPH is produced in which pathway? It is produced in HMP. The main pathway for NADPH production is HMP, right? But we have two minor pathways also. That is, one is malic enzyme, other is cytoplasmic isocitrate dehydrogenase enzyme, right? So these three things are producing NADPH, right? On the other hand, you have to see, other thing is NADPH is used in which pathway? It is used mainly for anabolic pathway, and this is known as reductive biosynthesis. NADPH is used in reductive biosynthesis, means for reductase enzyme in anabolic pathways, synthesis pathways, anabolic pathways, there it is required for giving hydrogen or reducing equivalence. So example of such pathways are fatty acid synthesis, cholesterol synthesis, steroid synthesis, and also in RBC for the conversion of oxidized glutathione to reduced glutathione. And you know, here we have two molecules, right? So these are all pathways where NADPH is used. This question is asking you about the use. So here only one option is correct, that is fatty acid synthesis. Answer is not all to this question. Be careful in reading the question, right? Now next question, mallet shuttle. It is about mallet shuttle. Mallet shuttle is required for Again, for this question, usually students have a tendency to mark all, but no, answer is not all, answer is glycolysis. Let's see the basics. What is mallet shuttle? Mallet shuttle is required for the transport of NADH from cytoplasm to mitochondria, because in mitochondria ETC occurs and NADH is the starting material of ETC, right? So we need NADH as starting material of ETC in mitochondria, right? But NADH as such cannot cross in a mitochondrial membrane, so we require shuttles. One of the shuttle is mallet shuttle, other shuttle is glycerol phosphate shuttle, right? So here the use is to transport NADH from cytoplasm to mitochondria. So can I say that any pathway which occurs in cytoplasm, only that pathway requires shuttle, right? So a pathway which occurs in cytoplasm and it is producing NADH like glycolysis, that require shuttle. But if a pathway like link reaction, pyruvate dehydrogenase reaction and TCA cycle, they are producing NADH in mitochondria only, because these pathways are occurring in mitochondria only. So NADH is already in mitochondria, there is no need of shuttles, right? So see the question again. Mallet shuttle is required for option A glycolysis, which is occurring in cytoplasm. Option B pyruvate dehydrogenase, which is occurring in mitochondria. Option C TCA, which is occurring in mitochondria. So answer to this question is A, glycolysis, the pathway which occurs in cytoplasm, that pathway requires shuttle, right? Next question, question number 6. The difference in molecular weight between phenylalanine and tyrosine is by. See, this reaction, you know that phenylalanine and tyrosine are aromatic amino acids. Phenylalanine is essential. Essential means we have to take it in diet. So from diet we get phenylalanine, and phenylalanine has only and only one role in the body, and that is it will form tyrosine. It will form tyrosine, and the enzyme here is phenylalanine hydroxylase enzyme. From the name hydroxylase, it appears as if we are adding OH group, right? We are adding OH group here in tyrosine, though you can see OH group is there, but actually what is happening, H was already there. H was already there in phenylalanine. The thing which is added is only one atom of oxygen, right? So these enzymes are also known as monooxygenases. Hydroxylases are also known as monooxygenases, because here only one atom of molecular oxygen is added. In the compound here, in the structure of tyrosine, you can see OH group, extra OH group, but H was already there in phenylalanine. The thing which is added is only O which is added. So the change in molecular weight will be by 16 only, not 17, right? Usually students have a tendency to mark 17 here as answer, but be careful, it is not 17, it is 16 is the answer to this question, right? Next question, question number 7. Ketone bodies are not used by which organ? Ketone bodies are not used by liver. See, there are two pathways for ketone body. One is ketone body synthesis, other is ketone body utilization or ketone body breakdown to get energy from them, right? Now, ketone body synthesis pathway also occurs in mitochondria. Ketone body utilization also occurs in mitochondria, but they occur in different organs, right? Ketone body synthesis can only and only occur in one organ, and that is liver. So it's like during the situation of starvation, now brain and heart needs ketone bodies as a fuel. At that time, liver has the responsibility of making ketone bodies, and only liver can make ketone bodies, and that is a vital thing for the body, right? So liver is making ketone bodies for other organs which will be using ketone bodies. So ketone body utilization or breakdown will occur in especially brain, heart, and also muscles. Earlier, this thing was a controversy that muscles can use ketone bodies or not, but now this thing is clear in your standard books that muscles can use ketone bodies, right? So, ketone body utilization is occurring in these three organs. So, what happens? That liver will make ketone bodies will come into blood, via blood they will travel into brain, heart or skeletal muscles, right? So, this is the scenario. Now, if liver is making ketone bodies, obviously liver should not use ketone bodies itself, right? Because liver is making it for others, right? So nature has made a plan that the very first enzyme of ketone body utilization, that is thiophorase, that is absent in liver, that is not given by nature to liver, so that liver can never use ketone body. Liver only has to produce ketone bodies, right? So answer to this question is liver, right?
Coming to question number 8, the question is all are functions of glycosaminoglycans except glycosaminoglycans. So let's take this topic in detail. Glycosaminoglycans are heteropolysaccharides. Heteropolysaccharides. Their short form is GAGs. GAGs, glycosaminoglycans. They are also called mucopolysaccharides. Mucopolysaccharides. They are also called proteoglycans. So there are 3-4 names I have written here. Now I'm going to explain each and every name. The first name is GAG, glycosaminoglycan. Why this name? Because the name says amino. Because there are lots of amino groups in GAGs. That's why this name. Then other name heteropolysaccharide. Why the name hetero? Hetero means different. Means they have different monosaccharide units attached to it, right? Then other name is mucopolysaccharides because they are slimy and slippery and therefore they are present in mucus secretions. That's why called mucopolysaccharides. Okay. And one more name, proteoglycans, because these gags are attached to proteins. These gags are attached to proteins making proteoglycans. All the gags are attached to proteins, but one exception is here. One exception, hyaluronic acid. Hyaluronic acid. It is a gag which is present in extracellular matrix. Most of the gags are present in extracellular matrix, right? So hyaluronic acid, when the structure of hyaluronic acid is there, then it is not attached to proteins. In fact, it is attached to other gags, right, in the extracellular matrix, right? One more exception here, I told you gags are extracellular, but one exception, heparin is a gag which is intracellular, right? Now let's see how gags are negatively charged. How they are negatively charged? They are negatively charged because of sulfate. They have lots of sulfate. They have lots of acid groups, so it ionizes to give negative charge. Then they also have lots of acetate groups. So see, mainly groups are present which are negatively charged, giving a negative charge to the full structure. Okay, now let's see the definition of gags. A very simple definition I'm going to tell you is they are tandem repeats. They are tandem repeats. Tandem repeats means one thing is repeated again and again, repeated one after the another. Okay, so gags are tandem repeats of two different things. One is AS, other is UA. I will tell you the full form. They are made up of two different things. That's why heteropolysaccharides. So that is AS and UA. So the structure will be like this. AS-UA is repeated again and again in tandem fashion like this. So n number of repeats and GAGs are linear. They're not branched. Usually students think that they are branched, but no, they are not branched. They are linear, right? And let's make the structure of GAG. I told you they are attached to proteins, right? So the full structure I'm going to make now. So first I am taking hyaluronic acid backbone. All the GAGs, when we make the structure of any GAG, then hyaluronic acid, which is one of the GAG, that is used as the backbone, right? So hyaluronic acid, which is one of the GAG, that is used as backbone. This is hyaluronic acid backbone. Then it is attached to core proteins. Many core proteins are attached to hyaluronic acid backbone, and here linker proteins are attached. This circular thing which I am making is linker protein. This thing which I made is core protein. I repeat, core protein is attached to hyaluronic acid backbone. Then these circular things are linker proteins which are attaching this core protein with the backbone, right? They are linking, so known as linker proteins, right? Now, these core proteins are attached to GAGs, the chain of GAGs, like this. Many linear chains of GAGs are attached here to the core protein, right? So, like this, on all the core proteins, like this. Can you see the structure? The whole structure looks like a bottle brush. So we say GAGs have bottle brush appearance, right? Now I want to zoom this area of core protein getting attached to the GAG backbone, right? So if I zoom that area, let's say this is the core protein. It is a protein. So it has amino acids present. So one of the amino acid side chain, that is serine. This is serine side chain is coming. It is the OH containing amino acid. So this OH of serine makes O glycosidic bond with the carbohydrate, that is the chain of GAG, right? But before starting the disaccharide repeats of ASUA, there is one core trisaccharide. There is one starting trisaccharide. So when this OH gets attached, then hydrogen is lost, and here we have xylose, galactose and galactose. Two galactose and one xylose here. This is a trisaccharide with the help of which linear GAG chains are attached to core proteins, and here I will write uronic acid amino sugar. You know, uronic acid and amino sugar are linear chains of GAGs. So I'm writing N here. There are many repeats of these disaccharides, that is amino sugar and uronic acid. So now one more very important point here is that this bond is a covalent bond. So I repeat that there is a covalent bond between core protein and the linear chain of GAGs, right? And there's a core trisaccharide which is joining the linear chain of GAGs with the core protein, right? So you can see the diagram here. When I make diagram, that is giving you a different kind of knowledge, and when you see already made diagram, that also has its importance. So I am showing you the diagrams also. So you can see here, this is chondroitin sulfate structure, and this green colored thing here is the core protein with which all the linear chain of GAGs are attached, and this is giving a bottle brush appearance, and this is a side view, right? If you view from the top, if you take a top view, then this kind of structure you will see, right? In the center, green color is core protein, right? Then you can see the examples of GAGs. Example of GAGs are like Cindycan, Glypican, right? Agrican. Now you can see Agrican here, biglycan, decorin and perlican. You can see some of the gags are cell surface gags, cell surface proteoglycans. This is cell membrane. This is cell membrane. And here you can see core protein. This is the core protein with which these linear chain of gags are attached, right? So glypican and syndican are cell surface proteoglycans, and we also have extracellular matrix gags. Those examples are decorin, perlican, biglycan, and egrican, right? This egrican is the main gag present in cartilages, right? Now let's see the examples of gags one by one. Gags. There are 6 gags I will be discussing. Number 1 is hyaluronic acid. Hyaluronic acid. Hyaluronic acid is present in vitreous humor, and synovial fluid, right? This gag is the longest gag. It is not having any sulfate. Usually gags have sulfate attached to them. That's why the names are heparin sulfate, chondroitin sulfate, dermatin sulfate, right? But see the name here, hyaluronic acid, it is not having sulfate. In the name also, there is no sulfate, right? So no sulfate here. Then this hyaluronic acid is responsible for cell migration during morphogenesis, right? Then it is also responsible for tumor cell migration, and it has role in wound healing. Recently this question was asked, which gag has a role in wound healing, and the answer is hyaluronic acid, right? Then coming to the second gag we are going to discuss is dermatan sulfate. Dermatan sulfate, which is present in skin, as the name says, derma, skin, blood vessels, and valves. Dermatan sulfate is most widely distributed gag. It is most widely distributed. This dermatan sulfate can be synthesized by arterial smooth muscle cells, and it binds LDL. Dermatan sulfate binds LDL, and therefore it has a role in atherosclerosis, right? Then the third gag I'm going to discuss is chondroitin sulfate. Chondroitin sulfate is the most abundant gag, as it is present in cartilages, bone, tendons, ligament, so these kind of places, and this is the most abundant gag, and relatively it is a larger molecule, right? This dermatan sulfate is a smaller molecule. It is widely distributed as it is present in skin, right? Then the fourth example is keratin sulfate. Keratin sulfate has no uronic acid. No uronic acid, a frequently asked question. Usually gags have uronic acid. All the gags have uronic acid, but keratin sulfate is not having uronic acid. And this keratin sulfate is of 2 types, keratin sulfate 1 and keratin sulfate type 2, right? Type 1 is present in cornea and it is responsible for corneal transparency. Type 2 is present in cartilages, right? And also keratin sulfate is most heterogeneous gag because it is having additional monosaccharides, like L-fucose, mannose, and NANA, N-acetyl neuraminic acid. So it is most heterogeneous gag. Let me tell you, in cornea or in eyes, 2 gags are present. Two gags are present. One is keratin sulfate, other is chondroitin sulfate. CS is chondroitin sulfate, right? Mainly keratin sulfate is present, but also some of chondroitin sulfate is also present, right? There is one mucin clot test. Mucin clot test, also called rope test for gags, but it is a non-specific test. I'm just going to tell you once here. What we do here is that we will add synovial fluid, add synovial fluid in acetic acid. In this test, add few drops of synovial fluid in acetic acid, and this will lead to the clot formation because of polymerization of hyaluronic acid, right? And this clot formation does not occur in few inflammatory diseases like rheumatoid arthritis, rheumatoid arthritis, gouty arthritis, and septic arthritis, right? But I told you it is non-specific test, so not of much use, right? Coming to question number 9, which of the following is a test to distinguish between monosaccharides and disaccharides? The test for distinction of monosaccharides and disaccharides is Barfoed's test. Barfoed's test is given positive by monosaccharides, right? Within 3, this is a reagent where we do the boiling, heating, right? Within 3 minutes, red precipitates are obtained, right? And this is monosaccharide positive, right? If you prolong the heating in this case, then up to 8 to 10 minutes, if you heat, then disaccharides also can give this test positive, right? So what we do to distinguish between monosaccharides and disaccharides, you do Barfoed's test for just 3 minutes, heat or boil just for 3 minutes. If red precipitates comes, then it is monosaccharides. If not, then it is disaccharide most probably, and then you can further heat it to check that it is disaccharides or not, because after around 8 to 10 minutes, these disaccharides will also give this test positive, right? So see here, this is the red precipitate which are positive for monosaccharides, or even this kind of picture you can see once. If image based question will be asked, you should be able to recognize the red precipitates of this Barfoed's test, right? Then let's do some more tests. One is Benedict's test. Benedict's test, which is for reducing sugars, You know that all monosaccharides are reducing. So they will give Benedict's positive. All monosaccharides give Benedict's positive, right? And only reducing disaccharides will give Benedict's positive, like lactose and maltose. You know that sucrose is a non-reducing disaccharide, so it will not give Benedict's positive, right? Then what about polysaccharides? Polysaccharides also do not react with Benedict's reagent because there is only one reducing end in a molecule of polysaccharide, right? So it is not reducing. That's why polysaccharides do not give this test positive. Now you can see the various colored tubes of Benedict's test. So various colors can come, right? And therefore it is a semi-quantitative test. Semi-quantitative test, that is, depending upon the amount of sugar, the color will vary, right? So the color tells us the amount of sugar here. See, Benedict's reagent is blue in color. The Benedict's reagent which we are using for the test, that is itself blue in color. So after heating, if blue remains blue, then that means it is negative, no sugar present, right? Then green, yellow, orange, and then red in sequence. I will help you learn this thing. How to learn? You know this Vibgyor, Vibgyor. This color spectrum Vibgyor. V and I leave them. Then B stands for blue, then green, then yellow, then orange, then red, right? Now, if blue color is obtained after boiling, then that says negative sugar, right? Green means 1 plus, yellow means 2 plus. So, in sequence, it is increasing, right? It is easy to learn this way. So, green means traces of sugar are present in the solution. Yellow means moderate amount of sugar is present. Orange means high amount of sugar is present, and red precipitates means very high amount of sugar is present. It gives an approximate idea of the quantity. So the quantity is like, if green, then it is 0.5 to 1 gram percent. If yellow, then 1 to 1.5 gram percent. If orange, then 1.5 to 2 gram percent. And if color is red, then it is more than 2 gram percent, right? So this way, Benedict's test is a semi-quantitative test, gives an idea of the quantity. Now, usually, frequently in urine, the reducing substance that is coming frequently in urine is glucose, right? But if some other reducing substance is coming in urine, which is non-carbohydrate, that can also give false positive Benedict's test. So, false positive Benedict's test can be given by non-carbohydrate substances which are reducing in nature. For example, vitamin C. If vitamin C is coming in urine in high doses, then it will give Benedict's positive even without boiling. You don't boil the solution, you just add vitamin C to Benedict's reagent, it will give positive, right? Then homogentisic acid, which will give black color to the solution because of oxidation of homogentisic acid, right? Then other substances which can give false positive test are uric acid, glutathione, ketones, creatinine, drugs like aspirin, cephalosporins. So this is a frequently asked question. You have to learn these substances which can give false positive Benedict's test. Now, usually we have to detect glucose in the sample, which is routinely done, and Benedict's test is not specific for glucose. So there are other methods for glucose estimation which you need to learn. There are two categories. Reductometric methods means reducing property of glucose is used. Other is enzymatic method, means some enzyme which break down glucose, that is used, right? So in reductometric methods, we have Nelson-Somogai method. You just need to see the names once. Nelson-Somogai, Folin-View method. These are not used methods these days, but you just need to know the name. And also orthotoluidine test. All these are methods which are reductometric methods for glucose. Then enzymatic method, one is hexokinase method, and this is the gold standard and very precise test for glucose estimation. The other method, enzymatic method, is GOD-POD method, which is the main routinely used method for blood glucose estimation. So we will do the detail of GOD-POD method here. GOD and POD, these two enzymes are actually used here. GOD stands for glucose oxidase, POD stands for peroxidase, right? Now, if the sample contains glucose, then first GOD enzyme will act on it and will do oxidation at carbon number C1, right, making acid, and that is known as aldonic acid, right. And also in this reaction, some amount of H2O2 is produced, which is directly proportional to the amount of glucose in the sample, right. Now this H2O2 is taken by enzyme POD, peroxidase, and this reaction gives a colored compound. Now, whenever we get some colored compound in biochemistry, then we have law of colorimetry, and estimating the color, we can estimate, by estimating color, we can estimate the amount of the analyte present, right? No need to learn the name of this complex colored compound, that is not required. You just need to know that in any reaction, if some color is formed, then we can measure the analyte, we can measure the amount of that analyte by using the law of colorimetry, right? So let's see some more tests in carbohydrates. This is the picture of Molish test, where a purple colored ring is the positive test, and Molish test is a general test for all carbohydrates. All carbohydrates, but there is a condition that number of carbons should be 5 or more. Number of carbons should be 5 or more. Otherwise, Molish test is not positive. For example, glyceraldehyde is having 3 carbon. Erythrose is having 4 carbons. These will not give Molish positive, right? Because they are less than 5 carbons, right? The other test is Selivanov's test, where red color comes. Selivanov's test, which is given positive by fructose, which is a keto sugar. So this test can distinguish between aldehyde and keto. See one more figure here. See, this is the red or maroon color, which is showing positive, means fructose is present, right? And see the other test tubes. This test tube is glucose, so it is not giving Selivanov's positive. And the third test tube, this is maltose solution. This is also aldehyde, so it is giving Selivanov's negative, right? Then one more test we have, that is iodine test, which gives black color as positive test, and iodine test means polysaccharide, especially starch, that is positive. So this is a potato where you know starch is present. So if you put iodine solution here, then it will give black color, right? And this black color is due to adsorption of iodine on the polysaccharide, right? Now one more test, that is osazone test, is given by monosaccharides and disaccharides, and these osazones are crystals which are formed only by reducing sugars, right? Now these are needle shaped osazones, which are for glucose, for fructose also same, for mannose also same shape of crystals, that is needle shaped, or we say broomstick appearance, right? Then the second one is sunflower shaped, which is for maltose. Then the third one is cotton ball shape, which is for lactose. Then the next one is for, these are rhombic plate like, and these are for galactose, right? So you have to learn the shape of osazone crystals for all these sugars, right? See one more diagram here. This diagram shows you very clear picture of these osazones. They are all mixed in this diagram. So here you can see these are like needles, needle shape. This is broomstick kind of needles joined together in a broomstick appearance, right? So you know this is for glucose. Then this is a typical sunflower shape, this area, sunflower shape, which is for maltose. Then this is a typical cotton ball shape. We also say hedgehog-like appearance. And this is for lactose. So you can see the clear picture here in this diagram, right? Now coming to next question, question number 10. How many ATPs are used during the transport of glucose via active transport? Active transport means energy is used. So students may think that 1, 2 or 3 ATPs will be used, but answer is 0 here. Be careful. So for that we have to do the detail of this glucose transport. So glucose transport is of two types. It is facilitative, which is done with the help of GLUTs, glucose transporters. On the other side, it is active transport, which is sodium glucose symport. Sodium glucose symport, right? Now, first we will do the detail of this active transport. So let's say this is a cell and this is sodium glucose symport here. Means symport means same direction, means both sodium and glucose are going inside the cell, right? Now what will happen when sodium is coming inside the cell? We cannot keep bringing sodium inside, because if you keep bringing sodium inside, then water will also follow and the cell will burst and die. So we can't do that, right? So every time we have to throw sodium out of the cell. We have to throw sodium out of cell. And how do we throw sodium out of cell? With the help of sodium potassium ATPase pump here, right? So energy is used by the sodium potassium ATPase pump, right? Energy is not used when glucose is entering into the cell, right? So when they ask this question, this is a tricky question. When they ask that via active transport, when glucose is coming in the cell, then how many ATPs are used? Here means they're asking here. So here no ATP is used, 0 ATPs are used. And this transport is using ATPs, using energy indirectly. So therefore it is called secondary active transport or indirectly acting transport, right? This secondary active transport is working against the concentration gradient. It is working against the concentration gradient. It is taking glucose unidirectionally. Only in one direction glucose can be taken, either inside the cell or outside the cell, right? Then this active transport is of two types. SGLT is the short form. S for sodium, Gl for glucose, T for transport. This is sodium dependent glucose transport. So we have two types, SGLT1 and SGLT2. SGLT1 is present in intestine and kidneys. It is for the transport of both galactose and glucose, right? SGLT2 is only present in kidneys and it is transporting only glucose, right? This is very important. SGLT1, SGLT2 you have to learn, right? So see this ready-made diagram here. Already I have made this diagram, but now you can see the ready-made diagram also. So this is kidneys where SGLT2 is present. This is renal tubular cell. This is the lumen here. This is interstitium here. You can see this sodium glucose symport SGLT2 is present here, which is taking sodium inside, glucose also inside. Both are going in same direction. And then sodium potassium ATPase pump is used to throw sodium outside, right? And glucose can be taken into the interstitial fluid with the help of GLUT2. Now this is the other form of transport, GLUTs, glucose transporters. or facilitative transport, which is the main glucose transport in body. See, SGLT is only used in intestine and kidney, nowhere else. But GLUTs are the main form of transport of glucose in body, which are present at various places, and there are various forms of these GLUTs, some of which are important. I'm going to discuss here. So see this table, which GLUT with the number, then second column is tissue location, where it is located, and the function of that GLUT, right? So first we will be taking GLUT1 and also GLUT3, because both these GLUTs are working in fasting condition in body. They are working in fasting condition. So during fasting condition, the channel should be open. It is a channel, it is glucose transporter channel, which should be open in places like brain, because brain always needs glucose, placenta. If the baby is there, then placenta needs glucose, whether mother is fasting or eating, right? Then kidneys for reabsorption work, then RBCs. RBCs also only they are dependent on glucose, that's why, right? So GLUT1 is present in brain, placenta, kidneys and RBCs. GLUT3 is also present in these kind of places, and here the thing written is brain, especially neurons, then placenta and kidney. Be careful, not in RBC. So in RBC, we have only, so if a question is asked, main GLUT present in RBC is, that is GLUT1. Okay. Now, as these GLUTs are working in fasting, so their function will be very simple, that is glucose uptake during fasting, right? Now, GLUT2, coming to GLUT2, which is activated in fed state, that is, as we eat glucose, GLUT2 will be opening, and this GLUT is present in intestine, so that glucose can be absorbed from intestine. So it is present in intestine and the function is absorption, right? Now, when glucose enters blood, then we have to store this excess glucose, and that occurs mainly in liver. In liver also this GLUT2 is present, which is active in fed state, so that extra glucose from blood goes to liver and helps in the formation of glycogen, right? So in liver the function is glycogen formation, right? Then in fed state, one more very important function is to release insulin, and that is done by pancreas. So a glucose channel in pancreas should also open in fed state, so that glucose enters into pancreas, and then pancreas will do its function of releasing insulin, right? So here GLUT2 is also present in pancreas and the function is insulin secretion, right? Now see here, see this diagram, this is pancreatic cell. When glucose is entering into the pancreatic cell, here glucose is entering, it will stimulate the release of insulin, right? So this way, from pancreas, in pancreas, GLUT2 is present, and when glucose enters via GLUT2, then there occurs release of insulin from pancreas. So now, when insulin comes, then insulin will activate GLUT4. Insulin will activate GLUT4. GLUT4 is present on peripheral tissue of the body, like muscles, skeletal muscle also, cardiac muscle also, and other peripheral tissue of body is adipose tissue. So here the function of GLUT4 is insulin stimulated glucose uptake after meals, because the work of insulin is to do hypoglycemic action, right? So it is like insulin will throw the glucose of blood into the peripheral cells, right? So that in blood we achieve hypoglycemic action, right? So can I say that GLUT4 is insulin dependent, and GLUT4 is responsible for hypoglycemic action of insulin, right? Okay, next is GLUT5. GLUT5 is for fructose transport, and it is present in small intestine for the absorption of fructose, and it is present in testis because sperms require fructose for energy, right? Then next is GLUT6. It is not very important because function is not known. You can just see it once where it is located. It is located in WBC and spleen. Otherwise, you can skip GLUT6 also, right? Next is GLUT7, which is present in endoplasmic reticulum of liver cells because it has a role in the transport of glucose from the endoplasmic reticulum, right? Then one more GLUT which is insulin dependent is GLUT8. There are less chances that these GLUTs will be asked, but you can just see them once. GLUT8 is present in blastocyst, testis and brain, and it is insulin dependent, and it can transport glucose, fructose as well as galactose, right? The next is last one, GLUT9 here, which is present in liver and kidney, and it is a urate transporter. It is urate transporter, right? So learn this table of GLUTs very nicely. It is a frequently asked topic in various exams. So let's quickly see these questions. Most widely distributed GLUT is GLUT1. Most abundant in RBC is GLUT1. Major GLUT in brain and placenta is GLUT1. In neurons, if they ask you in neurons, then it is GLUT3. If they ask you in brain both GLUT1 and 3 are present, but in neurons, GLUT3 is present, right? So GLUT3 is in neurons, but not GLUT1. Coming to next question, question number 11. Mutation in GLUT2 causes. Mutation in GLUT2 causes Fanconi-Bickel syndrome and one more variant, that is autosomal recessive proximal tubulopathy with hypercalciuria, right? So let's see this Fanconi-Bickel syndrome. Fanconi-Bickel syndrome clinical features resembles type 1 glycogen storage disease, that is von Gerk's disease. These patients have glycogen deposited in liver and kidney. They have fasting hypoglycemia, which is not responsive to glucagon, right? Then these patients are usually dwarfs. They have rickets. They have amino acid urea. They have phosphate urea, glucose urea, and postprandial. Postprandial, they have hyperglycemia. They have fasting hypoglycemia, but postprandial hyperglycemia, right? So this Fanconi-Bickel syndrome has many features similar to von Gerk's disease, right? Now let's see some mutations caused by glucose transport defect. SGLT1, SGLT2. If there is a mutation in SGLT1, this leads to glucose galactose malabsorption. If there is mutation in SGLT2, which is present in kidneys, then this leads to familial renal glycosuria, right? Then see the other options: Menkes disease, Beckwith syndrome, Dandy-Walker syndrome. I can just tell you one line for these three. Menkes disease is a condition of copper deficiency in body, right? Then Beckwith syndrome is overgrowth syndrome where patient has macroglossia, macrosomia, organomegaly, and some developmental abnormalities, especially abdominal wall defects. Abdominal wall defects like exomphalos. Then Dandy-Walker syndrome is affecting brain. These are a rare group of disorders affecting brain where cerebellum and fourth ventricle is mainly affected, right?
Next question, question number 12 is on Hartnupp's disease. It is a frequently asked topic. Hartnupp's disease is again autosomal recessive. Most of the metabolic disorders are autosomal recessive. Now Hartnupp's disease, in Hartnupp's disease, a transporter is defective. There is a transporter which is present in intestine also and present in kidneys also. This transporter is for neutral amino acids. Neutral amino acids, especially for tryptophan. Right. Now this neutral amino acid transporter is present in intestine for absorption, also present in kidneys for reabsorption, right? So this transporter is defective in case of Hartnupp's disease. When this transporter is defective, the body is unable to reabsorb that tryptophan from kidneys. So in urine amino acid urea, that is mainly tryptophan coming in urine, right? So now you should be looking at the uses of tryptophan, because here it's a situation of tryptophan deficiency in the body. So let's see the uses of tryptophan. One very important use of tryptophan is formation of niacin. Niacin is vitamin B3. See, this is a vitamin. Vitamins are usually not synthesized in body. We have to take them in diet, right? But can you see here, this vitamin B3 is getting formed in body from amino acid, that is tryptophan, right? So here this is known as atypical vitamin. Atypical vitamin which is getting formed in body, right? One more vitamin which is atypical is vitamin D. Vitamin D is also synthesized in our body from cholesterol under the effect of UV radiations from sunlight, right? So these two are atypical vitamins. When I talk of atypical vitamins, only these two are atypical, vitamin D and vitamin B3. Do not consider those vitamins which are synthesized by intestinal bacterial flora, like vitamin K synthesized by bacteria, intestinal bacteria, but that is not atypical. That is not synthesized by human enzyme, right? That is synthesized by bacterial enzyme. So there is a difference, right? Be careful. Okay. One more frequently asked question is, how much tryptophan forms how much niacin in our body? 60 milligram of tryptophan will form 1 milligram of niacin, right? Now, niacin deficiency is known as pellagra. So, can I say that in the patients of Hartnupp's disease, pellagra-like symptoms will occur? And this is your question. See the question. Symptoms of Hartnupp's disease mimic. So, answer is pellagra here, right? Now, coming to next question, question number 13. The following separation technique depends on the molecular size of the protein. Size or molecular weight is one and the same thing in biochemistry. So here your answer is gel filtration chromatography. What is gel filtration chromatography? It is also known as size exclusion chromatography. What we do here, it's like we have a flask in which a mixture of protein is there. Mixture of two proteins. We need to separate these two proteins by some chromatography. So what we will do, see the difference is in their size, right? So what we do, that we take a column, it is a column. In this column, we take gel molecules. These are gel molecules. This column is filled with this gel, right? The size of the gel is such that the bigger protein cannot enter the gel, the smaller protein can enter the gel, right? So now what will happen, when we will pour the mixture pour the mixture on the top of this column, then we have both proteins present here, bigger also, smaller also. Both will be coming down with gravity. Both will be coming down with gravity. But there is a difference that smaller protein, it will enter the gel, then leave, then enter, then leave, like that. Smaller protein is coming down like this. That is, it is getting hindrance from the gel. So it is coming down slowly, slowly, right? But see the bigger protein cannot enter the gel. Bigger protein will be coming down from in between spaces, and it will be coming down fast because it is not getting any hindrance from the gel. From in between spaces, it is coming down fast. So we say it is eluted first, separated first or eluted first. So bigger protein eluted first. After that, after some time, smaller protein will be coming downwards. We can collect these two proteins in different test tubes and we have done the separation, right? Earlier it was a mixture of proteins present. Now we have separated them with this technique where we are using gel, and therefore it is known as gel filtration chromatography, and we are excluding them on the basis of their size. So it is known as size exclusion chromatography, right? Now the gel which is used is known as Cefadex. The commercial name of this gel is Cefadex. Biochemically, it is dextran. Biochemically, it is dextran. What is dextran? I have already covered in a topic of classification of carbohydrates in homopolysaccharides, right? Now, the next question is a little tricky. Question number 14. Most abundant in bone. You know, in bone, mainly we have collagen. Collagen is having, you know, all the options are amino acids. So collagen is having which amino acid? It is having glycine, proline, lysine. Proline and lysine are hydroxylated, making hydroxyproline, hydroxylysine, right? So now you're confused between option B and option C. Which one to mark here? For that, you need to know that in case of collagen, most of the proline and lysine are hydroxylated. So if you have to select best out of these four options, you will mark hydroxyproline. So hydroxyproline is a better option to be marked as compared to proline. So this question checks this knowledge that you know that most of the proline and lysine are hydroxylated in collagen, and this hydroxylation helps in making hydrogen bonds, and too many hydrogen bonds gives strength to this molecule, this collagen, which is present in bones, and bones are carrying weight of the body. So this protein has to be very strong, right? Next question, question number 15. Mitochondria are involved in all of the following except. In mitochondria, ATP production occurs because TCA, ETC occurs in mitochondria. You all know apoptosis also occurs in mitochondria. TCA cycle occurs in mitochondria. But cholesterol synthesis occurs in cytoplasm. So answer to this question is cholesterol synthesis, right? So let's see what all other pathways occurs in mitochondria. Even link reaction occurs in mitochondria. Beta oxidation of fatty acid occurs in mitochondria. Vital pathways TCA, ETC occurs in mitochondria. Then both pathways of ketone body, that is ketone body synthesis, ketone body utilization, they all occur in mitochondria. Then in mitochondria we have mitochondrial DNA also, replication of this DNA. Replication of this mitochondrial DNA occurs in mitochondria. Also transcription and translation of this also occurs in mitochondria only, right? So all these pathways occur in mitochondria. Now next question number 16. Which of the following is active in phosphorylated state? Phosphorylation is done by hormone glucagon. Glucagon is active in fasting situation or catabolic situation in the body. So let's see which enzyme is involved in catabolic situation. Option A, glycogen synthase, involved in synthesis of glycogen. It is anabolic synthesis, right? So this is not the answer. Next, glycogen phosphorylase, rate limiting enzyme of glycogen breakdown, that is catabolic, and this is the answer to this question. See other options also. Next option, acetyl CoA carboxylase enzyme is rate limiting of fatty acid synthesis. This enzyme is also in anabolic pathway, so it will be active in dephosphorylated state. Then G6PD enzyme is enzyme of HMP, that is also considered anabolic pathway. So this enzyme is also active in dephosphorylated state, right? Next question, question number 17. Which of the following biochemical reaction is involved histidine to histamine? See, histidine is amino acid. Amino acid is getting converted to amine. Amino acid getting converted to amine. So only amino is left, acid is lost, that is lost in the form of carbon dioxide. Acid, COH group or carbon dioxide, we can relate, right? So on decarboxylation, histidine or any amino acid will be converted to its amine, histidine converted to histamine. So answer is this, decarboxylation. Very easy from the, if you are very clear with the words, with the basic things, then it is a very easy question, right? So let's take other examples of this decarboxylation. This is called simple decarboxylation, which requires vitamin B6. On the other hand, we also have oxidative decarboxylation, which requires vitamin B1 as coenzyme, right? This one is simple decarboxylation requiring B6. Other examples I'm going to take is tryptophan will be converted to tryptamine, tryptophan to tryptamine, tyrosine to tyramine. Then next is serine to ethanolamine, serine to ethanolamine. See, amine is written in all these names, right? You know why. Then cysteine gets converted to beta mercaptoethanolamine. So all these are examples of simple decarboxylation, right? Question number 18 now. Diabetic cataract is due to excessive accumulation of which molecule? You know, in diabetes, glucose is in excess, which will enter into insulin insensitive tissues like renal glomeruli, nerves, eyes like lens of the eyes and retina, right? So here glucose will be entering into peripheral cells like muscles and adipose tissue. Glucose cannot enter, but excess glucose enters into insulin insensitive tissues, that is those tissues where entry of glucose does not depend on insulin. In these cells, excess glucose enters, leading to the formation of sorbitol, and this requires enzyme reductase, that is aldose reductase enzyme is involved here, which forms sorbitol. Sorbitol also known as polyol. So because glucose is in excess, this sorbitol is in excess, which is responsible for giving snowflake cataract in diabetic patients, right? So answer to this question is sorbitol. Because of which substance accumulation cataract is occurring? It is sorbitol, right? So next question number 19, which isoenzyme of creatine kinase CK has least electrophoretic mobility? And this is the electrophoretic graph, CKMM, CKMB and CKBB. Let me tell you, electrophoresis is in this direction. This is CK1, this is CK2, this is CK3. Actually, when during electrophoresis, the isoenzyme form which is moving the most, that is given number 1, and the one which has moved the least, that is given last number. So least electrophoretic mobility is in CK3, that is CKMM, right? Similarly, in case of LDH also, 1, 2, 3, 4 and 5 we have. LDH, one isoenzyme will be having maximum electrophoretic mobility, and the last one, that is number 5, will be having least electrophoretic mobility. So it is very easy to tell if you know this basic point, that the one which has moved the most is given number 1. That's why it is given number 1. And the one which has moved the least is given the last number, right? So this question, which is not a biological derivative of tyrosine. They're asking about tyrosine. Tyrosine is an aromatic amino acid. It is aromatic amino acid synthesized from which amino acid? It is synthesized from phenylalanine. Phenylalanine gets converted to tyrosine in body, and therefore phenylalanine is essential. That is, we have to take phenylalanine in diet, but then in body phenylalanine can make tyrosine. So tyrosine is non-essential because we can make tyrosine in body, right? Now the enzyme for the conversion of phenylalanine to tyrosine is phenylalanine hydroxylase enzyme. And now this tyrosine has many biological functions in the body, and that is, number 1, it is used for the synthesis of catecholamines. Catecholamines, that is epinephrine, norepinephrine, dopamine, right? Then it is also used for the synthesis of thyroid hormones T3 and T4. And then it is also used for the formation of pigment melanin, right? So these are the main 3 uses of tyrosine in body. So let's see this question. Options are melanin, yes, pigment melanin is formed. Epinephrine, dopamine, they are catecholamines which are also formed from tyrosine. But melatonin is not formed from tyrosine. Melatonin is formed from tryptophan. It is formed from tryptophan. So both amino acids are starting from T, and both the products are starting from M, melanin and melatonin. So usually students get confused. So here be